So the next speaker is Minchole, who's going to talk to us about HPC for the investigation of the electrophysiological activity of the human heart.
Good morning. Thanks a lot for inviting me. My name is Minchole. I'm in the Computational Cardiovascular Science Group in the Computer Science Department at the University of Oxford. I will start by motivating the study of cardiovascular diseases. But the figures talk by themselves. 26% of all deaths in the United Kingdom and 31% globally are due to cardiovascular diseases, according to the British Heart Foundation. And there are a lot of research going on diagnosis of the diseases, mechanisms, but also on therapy. The first-line therapy of cardiovascular diseases is antiarrhythmic drugs. However, one of the most malignant side effects of these antiarrhythmic drugs is that they may lead to arrhythmias. So the questions here are why, which are the mechanisms for those arrhythmias, who, under which conditions these arrhythmias can take place, and also when. Those questions are still unclear, we don't know them, and there are many people working on them. We are investigating those questions from the computational medicine disciplines. This aims at developing quantitative approaches for understanding the mechanisms, diagnosis, and treatment of human diseases through computational techniques, but also mathematics, experimental physics, many things. So we work in cardiac modeling. We use multi-scale integration. We go from ionic currents through single cell, tissue, organ, going to body level, computing the electrophysiology at the body surface level, and ending with the simulation of the electrocardiogram. So at ionic level, let's try to use this. At ionic currents, these currents are modeled like ODs. Then at single cell level, those ionic currents are integrated in an action potential model that represent the electrical activity of a cell. And at these two levels, the investigations of the mechanisms can be done with a PC. However, when you include all this complexity and all these variables to whole organ, then to torso, and then you want to get to these higher levels of complexity, you need high-performance computing and also high-performance computing facilities. Archer, that is the one that we are using right now. Why HPC? As we said, at cellular level, the electrical activity is modeled by a set of ODs. Those ODs depend on the voltage, and this voltage is determined by the solution of a PDE. When we embed this into organ, we have to couple the cells. This is done by a PDE, and this is influenced by the heart geometry. But how a heart geometry looks like, or which are the... Here we have an example of a tetrahedral mesh of human ventricles coming from a CT scan. The edge length that we were using for simulating electrophysiology is 0.4 millimeters. It contains 2.5 million of nodes, each of them representing a cell. So then a cell model is in each of these nodes, and it contains also 14.2 million of elements. If we think that if we are using the latest human single cell model, that is the O'Hare-Ruddy model, it contains 40 variables. That means that we are dealing with about 100 million variables, that in double position is 800 megabytes for one time instant. So human cell model, this is an example of an action potential model, the electrical activity of a cell. It contains a lot of currents. Here they are the most important ones. And we observe that there is a large number of steep ODs. Some of them have rapid behavior, and other ones slower behavior. So it usually requires small time steps. And the most important thing for us is the computational power increases. And additionally, one bit, that is approximately 1,000 milliseconds, It contains, if we want to save every 1 millisecond, we are talking about 1,000 time instance. If we would like to save the information of 1 bit, it would be about 800 gigabytes. So these are big numbers, and HPC is necessary for them. Now I'm going to show you 3 examples in which we were using high-performance computing. This is the first one. We were interested in understanding mechanisms, and this is an organ-level simulation. In this one, our aim was to investigate and to simulate a heart attack and the effect of drug therapy. In a heart attack, normally there is a region of the ventricles in which is ischemic. Oxygen is not getting to that part of the heart. Here, this is the area, and it's surrounded by a border zone in which the appearance of ectopic activity that triggers a re-entry is higher. So this is our first model, and we were simulating the effect of these different drug doses to understand the mechanisms of the arrhythmia in each of the cases. Here we have an example. This is an activation of the ventricles. Then it comes the repolarization, that is here. Then it comes another beat, normal beats. This is the repolarization, and here it will appear an ectopic activity that will trigger a re-entry. So it surrounds the ischemic area and then gets in. In order to do this kind of simulations, we are using a parallelized monodomain model by finite element tetrahedral with linear interpolation. This mesh has 2.5 million of nodes with realistic cardiac fiber orientation. The action potential model that we were using is the 10-to-share model. It's a little bit simpler than the one I commented before, 19 state variables. And we solve it using the implicit backward Euler method. Using 1024 processors on the previous supercomputer of the United Kingdom, Hector, it took about 6 hours to simulate 800 milliseconds. And what's the discovery after all these things? Well, what we observed, we were investigating for different simulations. We were investigating the waveforms at whole organ level, and we observed that for high doses of this drug therapy, we were observing a lot of transmural reentries. That means that the waveform was coming from the endocardium and getting out to the epicardium. Here we have the second beat. The ectopic activity will come here soon. Here it is. It surrounds the ischemic area, and the reentry will come here, and then the arrhythmia is flowing. And here we will observe, like, we see this, that it's going to come from inside, from the endocardium to the epicardium, and we have several transmural reentries. This is what we observed in high doses of this drug therapy. And then, since these are multi-scale simulations, we were able to see what is happening in this area at this point, and we were observing this signature of the action potential. This signature is called early after depolarizations. Normally, they were observed in experiments, but never at whole organ level. And we observed, thanks to all these simulations, that electrotonically trigger early after depolarization, it was the ones facilitating the transmural reentries near the border zone under condition of high drug doses. Okay, this is another example. This is a body level simulations. In this one, we were not interested in the mechanisms. It was for diagnosis, for diagnostics, and what we wanted it was to simulate the ECG. The ECG is the electrical activity of the heart that is measured by some electrodes over the body surface. In order to do this, we were using a torsion mesh with lungs and ribs and the heart. It was 3.4 million of nodes, 19.4 million of tetrahedral elements. Different edge lengths were used, finer resolution in the heart, as imagine, and a much coarser mesh it was for the torso. 2048 cores distributed on 128 nodes were used, and a simulation of a heartbeat took about 1.5 hours. We were validating this model at whole organ, also using the body surface potential mappings and then the ECG. We already talked about the computational aspects, and once we have this model, we were investigating or simulating conduction blocks. These are the most typical ones: left bundle branch block and right bundle branch block. In the left bundle branch block, the left ventricle is not stimulated at the right time, it's a little bit delayed. The same happens with the right bundle branch block, in which the right ventricle is stimulated much, it's delayed. So we were simulating, these are the results of our simulations of this pathology, and these are the comparison with the patients diagnosed with this pathology for left bundle branch block and right bundle branch block. As we observe, they are quite similar, so the method is working quite well. And this is the last example I'm going to show you. This is the one we are working on. I mean, up to now, we were using the same torso, the same mesh for different pathologies, for different investigations, and here we were going towards personalization of models from MRI, magnetic resonance images, to ECG. So in order to do this, this is the pipeline we have developed. We start with standard cardiac MRI clinical acquisitions. We construct personalized meshes of heart, torso, lungs. Then we assign electrophysiological properties. We include fiber orientations. Eventually, we are going also to personalize the fiber orientations using DTI that we have for these patients. And we assign an activation sequence. And once we have all this, we are able to simulate electrophysiology at the body surface level and then simulate the ECG. So here we have the first step, that is, here are the typical 4 chambers view of MRI, 2 chambers, and short axis from base to apex. And here we have manual contours of right ventricle, left ventricle, endocardium, also epicardium, and with all this and some image processing, we are able to obtain personalized meshes of the heart of the patients. Also, we were using localizers. The localizers are not so high resolution images that they are taken in advance in order to locate the heart. With these images, we were delineating or segmenting different parts of the torso of the patient. This, together with a statistical model and regularization, taking into account the height and also the weight of the patient, we were able to personalize also the torso. So this is what we obtained, the personalized heart and torso and lungs for every individual. Once we do this thing, we need to construct our volumetric meshes, different resolutions, as I explained before, 0.4 millimeters for the heart, and we get to 0.8 centimeters edge length in the torso. And once we do this, we assign the electrophysiological properties, as I commented before, and we are able to simulate the electrical activity in the heart and then to the torso. And from here, and placing virtual electrodes to the patient, we are able to simulate the 12-lead ECG. Here is an example. This is the activation of the heart, the QRS. And here it comes, the repolarization, that is the T wave. This is our our first subject, and this subject contains, this mesh of the subject contains 44 million of elements, 7 million of nodes, and just the heart on its own, 5.2 million of nodes and 32 million of elements. A simulation of the electrical activity of 500 milliseconds using 960 processors in Archer takes around 6 hours. Sorry, and this is the subject, but we have already done 5 subjects. We observed that here we can study different orientation of the heart, different type of torsos. So this is what we are doing now. We have the heart of the different patients. As you can observe, the sizes are different, as all of us are different. Different torsos. Here we have the clinical 12-lead ECG that were recorded to these patients, and these are the simulated ones. The good thing with the computational, with cardiac modeling, is that you can fix all the properties. And what we wanted here is to evaluate the effect of the anatomy on the QRS, not taking into account possible differences from patient to patient in ionic properties or activation sequences that definitely we have. Each of us has different ones. We wanted to investigate just the effect of the anatomy on the ECG. And this is our work nowadays, mainly mine. And this is the final comments. HPC, high performance computing in cardiovascular science, were shown in disease, diagnostics, and also therapy. We demonstrate the power of HPC simulations for the investigation of disease, arrhythmia and drug effects in human electrophysiology, and personalized models of human hearts based on clinical MRI enable deeper understanding of the ECG. That's all. These are all the acknowledgements and many people that were part of this research. But thank you.